and welcome to our channel once more. We're happy to have you here, and as you know, our channel, that is Geo Academy YouTube, is biology and chemistry oriented, that is biochemistry. So you always find biology and chemistry contents in our channel. So today we're going to be working on a sequencing problem, or sequencing of polypeptides. So for today we're going to be sequencing, or we're going to see answer a question on the sequencing of vasopressin. So the question is right here before you, and it says, after treatment with peroxiformic acid, the peptide hormone vasopressin, in bracket a nonapeptide, is partially hydrolyzed. The following fragments are recovered. So the first fragment, you have phenylalanine, glutamine, and aspartate or aspartic acid. The second fragment, you have proline, arginine, glycine, and an amine. Now note this amine here. This amine here it does not mean it is the N terminal, but instead the C terminal is glycine, and the glycine has been ammonified. It means you reacted the C terminal with ammonia. So ammonia reacted with that C terminal with the carboxy group there to give this ammonia, and or this amide is actually something, an amide linkage, because water is given off since it's a condensation reaction. So the third fragment, you have cysteine, tyrosine, phenylalanine. Fourth fragment, asparagine, cysteine, proline, and arginine. Then the fifth fragment is tyrosine, phenylalanine, glutamine, and asparagine. Looking at the fragments, you see a similarity in that some amino acids occur in two or even more than two fragments. So we're going to see how we use that knowledge to answer the question. And there's also a very important word in the question, which is partially hydrolyzed. Partially hydrolyzed will serve us to answer this question equally. So the A part of the question says, what is the role of peroxiformic acid? So we move right away to answering our question. Peroxiformic acid, what is the role of peroxiformic acid? Okay, so we'll start by answering A. Peroxiformic acid, as you see here on the screen, is an oxidizing agent, and it will oxidize the disulfide bonds to form cysteic acid residues. So it cleaves the disulfide bonds actually, because the cysteic acid residues cannot combine back to form the disulfide bond. So it disrupts the disulfide bond by oxidizing cysteine to cysteic acid residues, and it equally is the agent that will partially hydrolyze our polypeptide. If you look at the question, I told you to note something on partially hydrolyzed. What does the partial hydrolysis is the peroxiformic acid. So it has two functions, oxidizing and partially hydrolyzing. So those are the two functions of peroxiformic acid. The question will have been asked maybe for 2 marks or so, depending on the person asking the question, or depending on the examiner in question. So that is all for the A part. We move to the B part of the question. So B is saying we should propose a structure for vasopressin, or we should propose the structure of vasopressin. How is vasopressin? How is it arranged? So since we know that the acid partially hydrolyzed vasopressin, and that it also cleaved the disulfide bonds. First hint to the question is that there is a disulfide bond. So a disulfide bond exists in vasopressin. Now, the second uh, thing to have in mind is that the 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 acid partially hydrolyzes the hormone and not completely. So that is why the fragments are some amino acids are common within the fragments. So we're going to use the knowledge of overlapping strands to answer this question. So those are the things to note to be able to answer the question. Now, right here before you is the main answer to this question. So when we match the overlapping regions, I told you regions that have common amino acids are overlapping. You can see this first strand here, cysteine, tyrosine, phenylalanine. Phenylalanine is also found in two more strands. In this second strand, it is found at the second position, right? So this first strand, it is found at the third. This second strand, it is found at the second, and this other strand, it is found at the first position. So you see that phenylalanine overlaps here in the three strands. Glutamine overlaps in two strands. Asparagine overlaps in three. Proline in two. Arginine in two. So that's the knowledge you use to bring out the sequence of this polypeptide or this hormone. So when you do like this, you bring out the overlapping regions, and if we are to count, you will see this is 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. No, sorry, that is not an amino acid. It is just an amine group. So there are 9 amino acids as expected. There are 9 amino acids in our hormone, because at the beginning they gave us a hint that it is a nonapeptide. So we are satisfied and we are rest assured that what we're doing is correct after counting. So from there we can bring out a complete structure of our amino acid, and you see that there are 2 cysteine residues. The first cysteine residue at position 1, the second is at 1, 2, 3, 1, 2, 3, 4, 5, 6, at position 6. So the disulfide bond formed is between position 1 and 6. Between position 1 and 6, you can see it right here in this complete structure of vasopressin that we propose after doing all this, and that is all about this sequencing problem. So this is all you had to do, just matching together the overlapping sequences or the overlapping regions and coming out with your complete structure of vasopressin or antidiuretic hormone. So that is all for the question, and see you in subsequent editions, as we'll be coming up with more questions and more content. See you. Thank you.